So the main topic is going to be fatty acid desaturase genes in Rosids. And it's a three-part talk, really. It's about the fatty acid desaturase pathway and genes and characterizing that in some species. It's the evolutionary story behind these genes. And then, if time permits, at the end, I'd like to give a very brief introduction of what I actually do with Mike in this department. So I'll talk about, let me just make sure, there we go. The fatty acid desaturase genes, what the pathways and genes are, the characterization work we did in cotton, the evolutionary history for these genes, and where this could go in the future. And you'll get to hear a bit about Waiuli, the highly heterozygous polyploid that Mike mentioned. So quickly, what are polyunsaturated fatty acids and why do we care? So for the plant, they're important as a response mechanism for cold temperatures. It helps protect the cellular function. For us, they're important for human nutrition. They're important oil seeds, seed oils, seeds, sorry. And synthesis of these oils occurs both in the ER and the endoplasmic reticulum and in the chloroplast. So it's an interesting biochemical pathway. There is traffic of components along the way between the two of them. It's a fundamental biochemical pathway. So it's interesting evolutionarily. It's present in all plants as far as we know. And there are only 7 genes really involved in this pathway. So it's a manageable pathway. It's really easy to work with compared to some of the other pathways. So what is fatty acid desaturation? Well, schematically, and for the biochemists in the room, I'm skipping a lot of details here. Using the 18-carbon chain fatty acids as an example, you get a source here in the ER, and then it goes through 3 stages, adding double bonds at each stage. And at each point, you can have exchange between the ER and the chloroplast, as well as for the final product. And this can be simplified even further to really just this 4-step process with a chloroplast set of enzymes and an ER set of enzymes. And the polyunsaturated fatty acids are these 2 products. So in the plant, you find a mix of these products, of these fatty acids. And the polyunsaturated fats are the ones that are important for cold tolerance and for human nutrition. In particular, the last one in the pathway, which are omega-3 fatty acids, which I think many of you have heard about. They're essential for fetal development in humans, and they're important for cardiovascular health. So it's not just an agronomically important pathway, it's also nutritionally important. So the focus of the talk today will be on the omega-3 fatty acid desaturases, specifically the genes that encode these enzymes at this point in the pathway. And they've been characterized in Arabidopsis as 3 genes. FAT3, FAT7, and FAT8 are the names in Arabidopsis. And there's one that encodes for enzymes functioning in the ER, and 2 copies that encode for enzymes functioning in the chloroplast. And functional characterization in Arabidopsis indicated that this particular copy is cold-induced. So when there is cold stress, the product from that particular gene is increased. So we wanted to expand that story and look in cotton. Why? Well, cotton is a tropical plant, so it's not well adapted to climates like Ithaca, or even anything north of Arizona, really. And especially for seedlings, even when you're planting it out in the desert, a couple of really cold nights can really damage your crops. So we want to see if cotton produces these fatty acids, how it produces it, how much and where. And it's also important for human consumption for the cottonseed oil improvement, the omega-3 fatty acids. So a brief introduction to cotton for those that are not familiar with it. Current cultivated cotton is down here, and it's a polyploid. And about 1 to 2 million years ago, there were these two ancestral cotton genomes that combined and formed a wild polyploid that eventually became the cultivated cotton. And at the same time, they continued their diploid species and genomes. So currently, we have this, which is cultivated, and we have these two wild cotton genomes. And this lets us put the cultivated cotton genomics in context. It lets us understand which of the two genomes is being used for what function by comparing them with the wild relatives. So we did an analysis of omega-3 fats in cotton. Specifically, we identified each homologous gene to the Arabidopsis genes in cotton and developed primers to separate the A genome copy versus the D genome copy and so forth. And what we're looking at here is cacao used as an outgroup. It's a closely related taxon to cotton. We have the A genome wild relative, the D genome wild relative, and then the cultivated cotton, which is a combination of both, but diverged for a couple million years from the wild relatives. And what we can see here is that for every one of the gene pairs, the D genome and the D prime genome from the polyploid, I'll use this as an example, grouped together. Each line points to one of the genes in this phylogeny. So they group together in the D group and the A group, suggesting that all of these copies were generated before the polyploid were formed. Every copy that's found in the polyploid, it's also found in the corresponding D genome or A genome. And what we have are really three taxonomic groups, each anchored by the cacao outgroup. So we have this group, which encodes for the ER-targeted genes, and we can tell that because of the targeting peptide. And we have these two groups that encode for the chloroplast-targeted proteins. And again, there is a D copy, both from the wild and the polyploid. There's an A copy, both from the wild and the polyploid. So all of this predate even the split of the A and D genome. So we can collapse them into 5 main genes, 2 for ER and 3 for the chloroplast. And we did functional characterization on these genes, essentially transcriptomics analysis in various tissues. And this has just been accepted in BMC plant biology, so it's in press right now. So we found that odd gene right there is a pseudogene. It has several stop codons in frame, and it's a specific duplication in the A genome. So we're ignoring that for now. It's not really useful functionally. We have one copy of the ER gene that's localized to cotton fiber in cultivated cotton. And it's by far the most highly expressed fatty acid dehydrogenase genes in cotton fiber. So it looks like this is a novel gain of function in cotton, but there is more work to really characterize that in detail. We found this copy that's cold induced in leaves, and this other copy that has fairly low expression in all the tissues that we've sampled. And these two copies out here without a label are constitutively expressed. So they're basically the basic functional copies of the gene that encode for the two necessary enzymes. So then we can put the story back together. We now added functional annotation for cotton. And from here on, I'm just using wild cotton as my sample, since the previous tree has shown that all copies are present in the AD genome, D genome, and the polyploid. So this simplifies the story. So we have the wild cotton functional representation and the Arabidopsis functional representation. And we can see a similar pattern. There is one constitutively expressed ER and chloroplast. There's a cold-induced copy, and cotton has a couple other duplicates. But what we wanted to understand is the origin of these gene copies. So we know that since they diverge, Arabidopsis and wild cotton have gone through independent whole genome duplications. 2 duplications in Arabidopsis, a triplication, and a duplication in cotton. So this sets up several hypotheses. Was there one gene or a couple of genes that were independently duplicated through those genome duplications in the 2 species? Or did all these 3 copies exist previously and cotton just added a couple new maintained copies? So to do this, we started with our Arabidopsis and cotton information, and we filled in with additional taxa without whole genome duplication. And all of these are sequenced genomes publicly available: papaya, cacao, grape, and cotton D-genome is also publicly available, and of course Arabidopsis. So these dates are very approximate. It's just to give you an idea of what the divergences are. And when I mention Rosids in the title, this is the broader clade. For those not familiar with the taxonomy, basically eudicots are split mainly into Rosids and asterids with a few other taxa. So this is a huge group of plants, diverged a long time ago. So given this sampling, we looked for the genes in the genome sequence, and we have these genes from Arabidopsis that we've identified previously. We have the genes we identify in our study in cotton, and the genomes of these diploids all had this pattern: 1 ER copy and 2 chloroplast copies. So this gives us a very nice answer to our question. The simplest explanation is that all 3 copies were present in the ancestor of this clade, and then the 2 copies in cotton were derived along this lineage, possibly due to the whole genome duplication. So this is a nice, clean story. In fact, very nice. I was surprised that every single one had identifiable copies of the genes. But we still have a lot of missing copies. If we have this hypothesis as the ancestral state, then given what we know about genome duplication, there should be 4 copies of each in Arabidopsis and 6 copies of each in cotton, because it triplicated and duplicated versus 2 duplications. And instead, what we find are 3 copies in Arabidopsis and 5 copies in cotton. So what happened to the rest of them? It's known in polyploids that there's a lot of genome reshuffling. Whole chunks of genome disappear, are lost after polyploidy. So it's possible that the whole region was just lost. But it could be that the genes themselves are missing. And to answer those kind of questions in the broader context of what happened in the genome rather than what happened to the copies, to the genes, we turn to Synteny Analysis. So briefly, we identified the genes in grape, which is our outgroup for this sampling. We then extend out to a larger region in the genome sequence for each of those 3 genes. And we selected that genomic region of about 750 kilobases in grape as our reference synthetic blocks. And we took that sequence and compared to every other genome sequence from the other species and looked to see if we could identify existing copies of those genomic regions. So now instead of looking at one gene, we're looking at a 750 kb genomic region. And that will let us, for example, differentiate between these 2 chloroplast copies because they'll be in different genomic contexts. The sequence of the genes themselves is fairly similar. So if you're looking at the generated annotation for many genomes, they'll be annotated as FAD7 or FAD8, depending on what it matched first in Arabidopsis. It's not very carefully annotated. So our hypothesis for the synthetic blocks now is the same, that there were 3 blocks of those regions before, and we expect this many copies in each of the taxa we're looking at. And when we did the analysis, and this was a lot of all versus all comparison of genomic regions and looking at conserved blocks and so forth. So I'm skipping over all of those gory details that I can talk to people who are interested about later. And this is what we found happened, or what we surmise happened. For the ER copy, we have all 4 copies identifiable in Arabidopsis, 1 copy identifiable in papaya and grape, and there was an inversion of that region. This whole block right here was inverted in the ancestor of cotton and cacao, and cotton has lost a small region for a couple of them, but we can find all of the ER regions, ER-related regions, the blue regions in all of the taxa. We can find all of the regions for Arabidopsis for this chloroplast, almost all for cotton and all in the diploids, and there are half of them missing for this region in cotton and 1 in Arabidopsis. So the genic regions are there, and we have this genomic context now, so we can put the genes back on. And we have the genes that we identify when looking at just the gene sequence. We have the genomic regions that we identify through Synteny, and we can add the functional annotation that we have for cotton and Arabidopsis on top of this. And this is sort of the big picture, bringing together phylogeny, Synteny work, and functional characterization work. And we have the cold-induced copy in cotton here, the low expression copy in cotton, and the constitutively expressed copy in cotton, plus the one that's expressed in fibers. We have the cold-induced copy in Arabidopsis. And you can see right away, these 2 are in different genomic backgrounds. Yet all 3 of the genomic backgrounds and the genes are maintained in the diploids, which to me suggests that there is a strong functional constraint for all 3 genes. So both copies of the chloroplast targeted one in particular suggest that are ancestrally functional. Whatever function they have, it goes back here. So that would suggest that the cold expression is an ancestral state in this background. But Arabidopsis doesn't have that copy. It only has a duplicate of the constitutively expressed one. So we looked closer at the Arabidopsis genes and the genomic regions, and we can tell actually which ones of these blocks were duplicated by which event, because they are fairly spread apart in Arabidopsis. And we can tell that the gene in this block was lost before this duplication. So this suggests actually a regain of function. And the functional characterization in Arabidopsis noted that the only difference between these 2 copies is in the last exon. Specifically, the exon in this copy promotes degradation of the protein except at low temperature. In other words, if the temperature is high, the protein from this copy is more degraded. So it's not an upstream control. It's not the cold expression in here is not controlled, not likely to be controlled by upstream regulators or anything like that. It's wholly contained within the gene region. So that's the story that we have so far from this analysis. And this is just a fairly small sample, but we've been able to at least hypothesize and set up for future work that these are likely dosage constrained. What that means is that more copies of the gene are likely to be detrimental. Even though so many regions are maintained in the polyploids, the copies of the genes themselves are reduced in Arabidopsis to just the 3 copies, and in cotton to 5. And the cold-induced phenotype is likely ancestral to at least the Rosids and shared by the Rosids based on the conservation of those regions and genes in all the plants sampled. And it appears that the gene in Arabidopsis is a newly recruited copy. So I think it's important to, if we're looking at a more generalizable control for the cold-induced gene, to look at species other than Arabidopsis. So where do we want to go from here? Well, this is a tree of all the fully sequenced genomes currently available. This is from Koji, genomeevolution.org. And this is current, well, it's probably out of date today. It's current as of last week. But there are a few genomes that are not here. And it's been an exponential increase in genome sequencing in plants. And you can see all of the polyploid events. Some are duplications, some are triplications, some are multiples. But the tree is rife with them. So from an evolutionary perspective, we have a lot of material to work with to look at the effects of polyploidy on genome evolution and on specific genes. These are the taxa we sampled. And those are the 2 taxa in which we have cold tolerance characterization. So what I'd like to see is broader sampling. I'd like to see something from legumes, from asterids, from grasses. We have no idea how this works in grasses. And I'd like to see cold-induced expression characterization in more taxa. Right now, we're making inference on cold response based on 2 samples. And they are rather different in how the genes, in the genes that are involved and so forth. So, everybody's mantra, more sampling, more experiments. I'm not going to be doing this. I'm working on completely different projects. So if there are any grad students that might be interested in any part of this project, come talk to me. And I'd be happy to discuss it with you. I wanted to put this out there for people to think about. It's an interesting system, and this pathway is fairly manageable, and we have the resources to expand this work. It's just a matter of hours and time, and lots of time spent staring at comparative Synteny maps. So with that in mind, it looks like I have time to switch briefly and talk about Wayuli, which is what I'm really working on in Mike's group. And the promise of Wayuli is an asterid, for those who are not familiar with it, is a domestic, sustainable source of natural rubber, if the plant feels like it. So Wayuli is a small shrub. It produces rubber, and it's one of only 2 other plants besides the rubber tree that are being investigated for rubber production. It's because of its habit, small perennial shrub, it's fairly amenable to mechanized agriculture and so forth. So it looks like a pretty good plant to develop as a crop. It produces noticeable amounts of rubber. And more specifically, why do we want to look at Wayuli rubber? Well, the US imports, as of a few years ago, about 3.3 billion worth of natural rubber, all coming from the rubber tree, which is essentially a monoculture in tropical areas. It's involved in deforestation because people burn down forests to plant it and so forth. Wayuli produces significant quantities of rubber, and it doesn't take a lot of work. Well, I'll put that in quotes because it's not really fully developed as a crop, so there are ways to go there. And it has been studied as a source of domestic rubber and intermittently cultivated for over 70 years, actually. So there is some history, there is some experience working with it. However, Wayuli has a very fascinating mating system. It has variable sporophytic self-incompatibility, and the variability is among accessions and populations, and more importantly, between ploidy levels. The diploids are completely incompatible. The polyploids vary as to the degree of self-incompatibility. It's a facultative apomix, and for those familiar with apomixes, I've listed the details there. But to summarize the take-home from the facultative apomixes and self-incompatibility, we have this problem. It needs cross-pollination to set maternally clonal seed. It needs pollination to set viable seed because the endosperm needs to be pollinated. But in most cases, the genotype of the progeny is maternally clonal. So, I'm sure every breeder in the room is shaking their head right now at that. And moreover, both of them are facultative or variable, so they can't be relied upon for breeding schemes. You can't just plant accessions next to each other and say, well, they're apomix, they'll never cross. Because people have tried to disastrous results. And because of the facultative apomixes system, you have 4 possible progeny types. Apomixes, for those not familiar with it, it's really 2 parts. There is the meiotic reduction or non-reduction of gametes, and then there is the parthenogenesis part, which sets seed. And in the case of Wayuli, you also have fertilization or no fertilization. So if you have fertilization and non-reduction, you get a maternal genotype. If you have no fertilization and no reduction. And this is the classical apomix that people assumed was the system in Wayuli. If you have reduction and no fertilization, you end up with polyhaploids. So if you have a tetraploid that has that happening, you end up with something that looks like a diploid. And that's not uncommon, actually, in Wayuli. It's been observed in the literature. Then for non-reduction and fertilization, you have changes in ploidy level. In fact, in both cases, the ploidy change depends on the pollen donor. The pollen is always reduced. So you have clonal offsprings with little genetic diversity from 2 of the cases, sexual offspring, ploidy bridges. It's a mess. This all happens at the same time in the same population, and sometimes across species in Parthenium. So because of that, because of the ploidy variability, the ploidy and the genome size vary. And I have this a plot from a paper we published this year, I guess. So this plots the chromosome counts. We actually did chromosome squashes and counts for several samples from several Parthenium species. The gray here, it might be a bit hard to see, is Wayuli, Parthenium argentatum. And here are the diploids, triploids, tetraploids, pentaploids, and hexaploids. So it just keeps going. And you can see there is also aneuploidy going on. So where only some of the chromosomes get duplicated. So instead of a whole doubling, you have 1.5. Or some of them are lost after duplication. And that's going on. These are, again, chromosome counts in between what we expect the duplicates to be. And we have genome size variation. That's one species in Parthenium that's twice the size in genome of the corresponding same chromosome number other species. So there's a lot of interesting work to be done here. Actually, I find it very exciting because I get to look at ploidy series and figure out what's going on. We have variable incompatibility and variable apomixes. So with a large enough sampling, we could probably try to tackle the question of which one is the apomixis locus and split it into parthenogenesis locus versus non-disjunction and so forth. And for our project, we are sequencing, genotyping by sequencing the entire USDA germplasm of Waiuli. So there are about 80 to 100 accessions roughly. And we're doing several samples per accession. And we are doing a genome sequence of the diploid Waiuli. So we'll have the tools to address these questions. So I'm excited to dig into this highly heterozygous multi-polyploid that does whatever it feels like it whenever it feels like doing it. And I'd like to acknowledge the people that are involved in all this work, the Gore Lab in general for lots of helpful simulating discussions and work in Waiuli. John Dyer, Olga, and Joshua, you all did the work in the characterization of the functional characterization of cotton. And the cotton work was funded by Cotton Inc. This grant funds the Waiuli work and Cornell in general for supporting all of us. So with that, I want to thank you for listening and take any questions.